this national conference. This session will be chaired by Professor Giridhar Vedantam, Principal Guru Gorakhnath University of Medical Science, MAHAYOGI Gorakhnath University. We welcome you, sir. Thank you, sir. Co-chaired by Dr. Atul Rukadikar, Associate Professor, Department of Microbiology, All India Institute of Medical Science, Gorakhpur. We welcome you, sir. Thank you, sir. Assisted by Dr. Dhirendra Kumar Singh as rapporteur. We welcome you, sir. Thank you, sir. We extend a warm welcome to our distinguished panel and invite all the participants to engage attentively the session. It is my privilege to invite our first speaker of this session, Professor Dr. Shailendra Kumar Singh, Department of Microbiology, Dr. Ram Manohar Loya, Awadh University, Ayodhya, to deliver his lecture on the topic of environmental dissemination of antibiotic resistance to hospital effluents. Professor Kumar, may we kindly request you to take the stage.
Thank you for inviting me here on this conference. So this is a privilege for me to be here among the young students related to the discipline of microbiology. As the conference has been organized to dedicate the antimicrobial resistance. So I think it is good to talk about the antimicrobial resistance here. Many more topics we can cover elsewhere. So the talk of mine is to be based on the environmental dissemination of antibiotic resistance through hospitals.
So it is my privilege to introduce today's speaker, Professor Shailendra Kumar ji. Professor Shailendra Kumar is Professor of Microbiology at Ram Manohar Lohiya, Awadh University, Ayodhya. He is having very good expertise in molecular drug resistance, environmental microbiology, and green synthesis of nanoparticles. His research is on integration of fundamental microbiological approaches with applied innovations, contribution to sustainable solutions in healthcare, environmental management. Professor Kumar has published extensively in reputed international journals, authored many books on microbiology and biotechnology. And he has been honored with many prestigious honors, including Distinguished Scientific Award, Excellence in Teaching Award, and by Society of Agriculture Innovation Development, SAID Ranchi, and Young Scientist Award from Indian Council of Food and Agriculture. So, sir is actively engaged in advancing molecular diagnostics and nanotechnology-based approaches and addressing present biomedical challenges. So, his works on drug resistance, nanoparticle synthesis are real-world applications. So, with this, we are honored to have you, sir, here for the lecture, and I request you to continue with the lecture. Thank you, sir.
Thank you, sir, for giving such a nice introduction about me. So Antibiotic resistance develops only after we are introduced to the antibiotics, right? Agreed with or not? Unless we are exposed to the antibiotics, the antibiotic resistance may not develop. And how old the antibiotics are, do we know? Since the discovery of the penicillin in 1929 by Alexander Fleming. So what about the history before the reports by Alexander Fleming? So you can notice that traditionally it is not exactly the microbiology product, neither the antibiotics, but something related to it has been used. Like the Chinese people, they used moldy soybean curd to treat the boils on their foot infections. They used to wear the sandals which are furry with the molds, and that controls the foot infections. That was the therapy which they were taking. Before 1929, Kendall introduced about the clearance of the cloudy substances which are growing into the bacterial culture medium, that is cleared by the development of the mold on the surface of the medium. Several other researchers, Pasteur and Jobert, they demonstrated the growth of anthrax in urine. Emmerich and Lowe, they described about the thiocyanase, that is also a kind of antibiotic, but they did not call it antibiotic, neither they used to use the compound as an antibiotic. So before the development of the antibiotics, the replacement therapy was proposed by Mechnikov in 1909, for which he received the Nobel Prize also. Garcia and Dark, 1925, they introduced about the actinomycetin. And then comes the work of Alexander Fleming, which is addressed as the father of antibacterial substances, Alexander Fleming, who observed that Staphylococcus aureus, which is growing on a petri dish, that was inhibited by the growth of a mold, fungi. And that fungi was identified as the Penicillium species. अब 1929 में Alexander Fleming ने report कर दिया, but still nobody was ready there to accept that as a compound penicillin. Simply because our medicine people, they do not just jump on any therapy. First, they need to do certain number of studies to believe that what is being recommended, that is correct or not. And at that time, when penicillin was discovered by Alexander Fleming, the more the impact of the chemical drugs was there. They were in high use. Arsenic compounds were used. Salvarsan compounds were used. And azo compounds were used. So use of any of such kind of antibiotics, which is a produce of the microorganisms against the treatment of the, for the treatment of the antibacterial, as an antibacterial agent, it was out of course. So nobody was relying on this. And René Dubos, they isolated a bacteria brevis, Bacillus brevis, and they demonstrated that the cell-free extract of the bacteria, that is to inhibit the other bacteria. And the compound which it is producing is gramicidin and tyroside. Selman, Waxman and associates, they demonstrated the streptomycin. And only after 1945, the use of the antibiotics started. And that too was very rare or very small. Rarely the antibiotics were used. We are using the antibiotics since 1960 or 65. Before that, it was all out of course to use the antibiotics. And these are the mechanisms by which the antibiotics work. I think more the details have already been described to you. In the morning session also, Madam told about the mode of infection of the mode of action of the antibiotics. Like briefly, I can just say it here, inhibition of cell wall synthesis. You all know what is the cell wall of the bacteria. Bacterial cell wall is composed of peptidoglycan, right? Peptidoglycan is the main component of the gram-positive bacteria, and gram-negative bacteria is having the peptidoglycan, but very thin layer of the peptidoglycan, right? And peptidoglycan provides strength to the cell. It makes the shape of the cell. If the peptidoglycan is not synthesized properly or is inhibited, the bacterial cell wall will be disrupted, right? So if you stop the bacterial cell wall synthesis, that is going to develop a bacterial cell without cell wall. Is that possible or not? Yes, that is how you can develop the PPLO. If you put the bacteria into a very sublethal concentration of the antibiotics, it will develop the bacterial cell devoid of the bacterial cell wall. Only the cell membrane will be there and the cytoplasm containing the other organelles. Damage to the cytoplasmic membrane. Certain compounds are there which are to damage the cytoplasmic membrane. They will create the pores into the cytoplasmic membrane and the bacteria will leak out. Likewise, the inhibition of the nucleic acid and the protein synthesis. Several antibiotics are there which are acting upon the DNA gyrases. They cleave the DNA, right? So they interfere with the cleavage and ligation mechanism of the DNA, which is a compulsory part of the DNA replication. Likewise, the protein synthesis is also very essential. The protein synthesis is an essential component for the cell survival. Now you know that if the protein synthesis is blocked, what will happen? The cell is to lose its life. Simply because the protein is not being synthesized, the structure will be lost. As well, certain number of functions which are dependent upon the proteins, that will also be inhibited. Inhibition of a specific enzyme system. So these are the various mechanisms by which the antibiotics work. Now it is the term of the bacteria to overcome these mechanisms. So how do they overcome? Definitely they have to have work smartly better than the antibiotics. So if there is an antibiotic which is going to inhibit peptidoglycan synthesis by binding to an enzyme called transpeptidase. Transpeptidase पढ़ा है ना आपने? पानी दीजिएगा तो. अच्छा. Okay. So peptidoglycan layer, there is an enzyme called transpeptidase which will make the dipeptide, right? Two short peptides, they are brought together to make a peptide bond. Transpeptidase enzyme is much required to provide the stability to the peptidoglycan. So if a bacteria is there which can resist the antibiotic penicillin, how will it do? It will have to produce an enzyme which can act upon the penicillin compound and degrade it, right? So they have to have a penicillin binding enzyme which binds to the penicillin, cleaves it, and the effect of the penicillin is gone. Likewise, those antibiotics which are going to act upon the nucleic acid synthesis or the protein synthesis, their target site is modified. How is it modified? Definitely, these are mostly the proteinaceous substances which are attacked upon by the antibiotics. So they have to have some mutations, some conformational modifications in the protein, so that the antibiotics will not get a space to bind to these sites and they overcome. So if the bacteria is smart, it is changing itself so that it can overcome the effect of the antibiotics, right? That leads to the development of the antibiotic resistance or antimicrobial resistance. It has been classified that if a bacteria is resistant to a single antibiotic, it is called antibiotic resistance. If it is resistant to single antibiotic, it is antibiotic resistant bacteria. And if it is resistant to more than one antibiotics belonging to the different classes, and up to 3, we can call it multi-drug resistant bacteria. There is a group of the bacteria which are able to degrade or able to overcome the antibiotics across the various classes. That is called extremely drug resistant, and further to that, the pan-drug resistant. So you can imagine it is hardly about 50-60 years since we have been using the antibiotics, and we are approaching the time when there are the microorganisms which are resistant to not one only but almost all the classes of the antibiotics. Then where we are going to lead? What is the future? How we will survive without the antibiotics? Because more or less we are dependent upon the antibiotics like anything else. So how this antibiotic resistance is developing? To do an assessment, we tried to carry out a study on the hospital wastewater. You know, the hospitals are the sites where the patients are admitted. They perform their daily activities because they are admitted there. Whatever they are consuming, the drugs that is being discharged, that goes into the drain. And not only that, but in fact, other than that, the drugs which are being used in the hospitals, that is also being disposed of through the drainage system. The hospitals are the sites from where you can expect to get the bacteria which may be resistant to not only one or but several drugs. So multiple drug resistant bacteria or pan-drug resistant bacteria or extremely drug resistant bacteria, this all is very common in the hospital settings. And how these organisms are being discharged into the water system. So if this water is carrying the antibiotic resistant bacteria and is merging with the other larger bodies, then what is to happen? The possibilities are there that those organisms which are sensitive to the antibiotics, they will also acquire the resistance. As we all have studied, the mechanism of the acquirement of the resistance, there are several ways you have studied. How do they develop the resistance? Two factors are there. One is called intrinsic factor, another is extrinsic factor. So intrinsic factor is when the bacteria has itself the resistance mechanism from its own gene, either located on the plasmids or on the chromosome. And extrinsic factor is acquired factor, that how the resistant organisms, they can transmit their gene to the sensitive organisms. So several mechanisms are there. Conjugation is there, transduction is there, transformation is there, by which they can acquire the genetic elements and develop the resistance, right? So if the resistant organisms are being discharged into the hospital wastewater, and that is leading to the water bodies where it can meet with those microorganisms which are sensitive to the antibiotics. Now the sensitive microorganisms can also develop the antibiotic resistance. So with this aim, we tried to carry out this study. As this all has been already informed to you that this AMR is one of the top 10 goals which have been decided by the WHO, that these are the top health problems of the world. And why is it top health problem? It is because 4.95 million deaths have been associated with the AMR by 2019. And if we do not take care, and the way it is going on, if we keep it, let it go, then it is to touch 10 million deaths annually by 2050. And what will be the condition? No cure against these infections which are caused by the multi-drug resistant or pan-drug resistant organisms. The economic and societal impact, that is also very high. About 24 million people into extreme poverty by 2030. And if it is not regulated, the loss is to increase. Water is the system by which the dissemination is to occur. So this study was planned to carry out the physicochemical and microbiological analysis of the water samples which are collected from the various drainage sites of the hospitals, isolation of the microorganisms, and then we carried out the determination of the antibiotic sensitivity profile or the resistance profile of these microorganisms. And one parameter is there that is called multiple antibiotic resistance index. That helps us to do this assessment that if there is a resistant organism available in a reservoir, then what is the possibility of it to transmit the resistance to other organisms. That has been developed a technology to do this, calculate the MAR index. I will share with the results. This is the sites from where the sampling was done. A total 9 different hospitals located in Ayodhya were selected out for the purpose, which were one of those hospitals. They have the high inflow, including the government hospitals. So what we found, the pH of the water is 6.4 to 7.7. This is within the recommended limit as is prescribed by WHO or CPCB or other authorities, APHA. The temperature is also 20 to 22 degrees. So you can say that no hot water is flowing through. Elevated TSS, total suspended solids, 180 to 380 milligram per liter has been reported. That exceeds almost 5 times the recommended limit. That means the water which is flowing out from the hospital that is having certain suspended solids. The BOD and COD level is also high, but in our cases, what we have reported is not that high as has been reported by the larger cities and the developed countries. Though the condition is not good here also, the BOD 50 to 69 milligram per liter, COD is 204 to 288 milligram per liter. It should be even lower, almost 10 times lower than what has been reported here. The low dissolved oxygen content is seen there, 0.2 to 1.87 milligram per liter. So this table shows the same data which was summarized in the previous descriptions. The microbiological analysis was performed. The bacteria were isolated from the water samples and they were identified, characterized as 179 isolates belonging to the different of these genus species: Escherichia coli, Klebsiella, Shigella, Salmonella, Proteus, and Pseudomonas. Actually, it may have certain other microorganisms which belong to other families, but our focus was because the waterborne diseases, mostly they are carried out by the Enterobacteriaceae members. So our aim was to see whether the Enterobacteriaceae members are there, what are the contents, and whether they are acquiring the resistance or not, because more the chances to acquire the diseases from these microorganisms, they are the intestinal infections if you consume the water. So these bacteria were identified by the biochemical tests. Their antibiotic sensitivity profile was carried out. The method which has been used, you all must be doing in your lab, Kirby bar method. Prepare the media, Muller-Hinton agar, place the bacterial culture, and then put the discs containing the antibiotics, right? And then place it into the incubator for the growth. The bacteria is growing, and if it is sensitive to the antibiotics, then there should be no growth around the disc. So you see here, 7 antibiotics in one petri dish, and 7 in other, and no zone. That is the situation we reported with. None of the antibiotics were capable to develop the zone against these bacteria. So certain, this is the sensitivity profile. You see here, Pseudomonas species there, which is 100% resistant to beta-lactam. Likewise, it is resistant to macrolide also. These are very advanced drugs and are considered to be the safest drugs. So if this is the condition of the safest drugs, what will happen to those drugs which are very commonly used? This profile was analyzed for the classification of the MDR, XDR and PDR. MDR is more than one, but around 3 antibiotics are being resisted upon by the bacteria. XDR is when about 10 antibiotics are not working on the bacteria. And PDR is when no of the antibiotic is working. You see here, all the 3 classes are reported. The highest is the MDR, then XDR, and out of 179 bacterial culture, we got 3 bacteria which were resistant to all the antibiotics. MAR index, that particularly helps us to do this assessment that whether the water body is secure or safe, or there are chances for the antibiotic resistance development. And that is calculated if the MAR index is below 0.2. If the MAR index is below 0.2, it can be said that there is no chance of the development of the antibiotic resistance. But in this case, all the samples of the bacteria which were isolated from the water sources, the MAR index is very high, even up to 0.65. That means it gives an indication that these water sources are very much capable to transmit the resistance features to those organisms which are sensitive and are reported into the same reservoir. So here we see that the physicochemical features, that is not very say impactful here, but still the antibiotic resistance profiles of the antibiotic resistant bacteria, PDR, MDR, XDR, that is alarming, and it gives us a signal to do something. Otherwise, it will be too late to have the dependency on the antibiotics. There is an urgent need for the management of the wastewater. How do we manage? How do we do the treatment of the wastewater which is going out from the hospitals? It is very much required, because if we do not do this, then in turn what is going to happen? This water is going to merge with the water bodies, and the MAR index values, they are suggestive of the development of the antibiotic resistance at very alarming rate. And then what has been assumed by the WHO by 2050, the death rate will go to more than 10 million per year due to the antibiotic resistance. So, and we all know what is the reason for this. What do you assume? What is the reason? Yeah, it is the use or misuse of the antibiotics, you can say, for which the government also brings out the derives. Antimicrobial Awareness Week has been celebrated recently in the month of November. So this is going to help. I think so. But no, this awareness week is not going to help anyway. Simply if we do not change our habits and the dependency, like अभी आप में से बहुत सारे स्टूडेंट्स ऐसे बैठे हैं यहाँ पे जिनको थोड़ा सा कुछ प्रॉब्लम होता है, सर्दी हुआ, जुकाम हुआ, हल्की सी खांसी आई, तो क्या करते हैं वो? They do not go to the doctor. They will simply go to the medical stores and buy the medicines on their own. Either they have it in their box also. Azithromycin. Azithromycin has become a very common drug, which is used like as if you have some chocolate in your pocket. If you ask what happened, sir, I'm having sore throat and coughing for 2 days. So have you taken some medicines and visited the doctor? No, I didn't visit the doctor, but I had azithromycin from my past treatment. So I used that. Do you know how many days and what the doses should be of the azithromycin? That I don't know. How many times you should take and how should you take, when you should not take, nothing is known. But it is the azithromycin which will work if you have some cough. Likewise, people do the treatment of their own, for which the antibiotics are classified under Schedule H drug. Right, sir? And Schedule H is a schedule which does not allow the medical stores people to sell the medicine without a prescription letter. But here, nothing is banned. One question comes that whether the development of the antibiotic resistance is really dependent upon the use of the antibiotics. So with this, a study was performed to find out those microorganisms which have never been exposed to the antibiotics. Like you visit the tribal areas or the deep into the forest, isolate the bacteria and perform the antibiotic resistance or sensitivity test. So what do we expect? We expect that those bacteria will not have any kind of resistance. But this is also not right. The bacteria isolated from totally a new place which has never been exposed to the antibiotics, they may also have resistance. And they have resistance because it is a kind of natural phenomenon which is going on. You have studied about the microorganisms interactions, microbial interactions in the environment. So there are microorganisms. We are deriving the antibiotics from the microorganisms. We are using them against the microorganisms. In the nature also, those microorganisms are present. They are producing the antibiotics, and those antibiotics are exposed to the other microorganisms and they are developing the resistance. It is a kind of continuous evolution which is going on into the nature. Yes, definitely. But what we do, our malpractices about the uses of the antibiotics, that is affecting the development of the resistance, that is helping the development of the resistance too quickly. Means the resistance which could have been attained in the nature maybe some 50 or 100 years later, that we have brought very close to end. And which could have happened in 2300 years, now that is happening in 2025-26. You isolate a bacteria from anywhere. We have the practices to throw away the medicines whichever is left over at home. We throw it into the dustbin and dustbin takes to the landfilling sites, which in turn is making the way to the water bodies. The water body is getting contaminated with the antibiotics and the antibiotic resistance development. It's very normal mechanism of the development of the resistance. So only about the only making awareness about the uses of antibiotics, that how should we use, how to use and how not to use, what to do with the antibiotics whichever is left over, that is also very much required. so should not be eaten like the snacks. Thank you very much for listening me so patiently. I thought of that after lunch it is really difficult to keep you alive because mostly the people they fall asleep. But I think you were not sleeping. Are you awake or asleep?
So, very good afternoon. And first of all, I would like to appreciate your presentation on this environmental dissemination of antibiotic resistance through hospital influence. So, आपने सही मायने में बताया कि अगर हम बराबर एंटीबायोटिक्स नहीं यूज करेंगे या ओवर यूज करेंगे, so there might be a chances कि we will be left with no antibiotics. अभी के अगर आज का सिनारियो देखा तो इट इज ओनली 2 एंटीबायोटिक्स विच आर बीइंग लेफ्ट, जिसमें ऑर्गेनिज्म ने ऑलरेडी रेजिस्टेंस दिखाया है, बट इट इज टू सम एक्सटेंट एक्सेप्टेबल, एंड दैट एंटीबायोटिक इज कोलेस्टिन. तो ये कोलेस्टिन ड्रग जो है वो जैसे सर ने बोला कि जैसे पेनिसिलिन इन्वेंट हुआ था तभी 1948 टू 50 में इन्वेंट हुआ था. दैट टाइम इट वाज नॉट बीइंग यूज्ड टिल डेट. उसकी वजह से वो एंटीबायोटिक बच गया. बट नाउ सिंस वी आर लेफ्ट विथ नो एंटीबायोटिक्स, द कोलेस्टिन जो लास्ट रिजर्व ड्रग है वो लाया गया, बट अभी भी उसको अभी रेजिस्टेंस डेवलप हो गया है. सो ऑलरेडी सर हैज मेंशन कि व्हाट इज द मेन फैक्टर एंटीमाइक्रोबियल रेजिस्टेंस का, तो इनजुडिशियस यूज ऑफ एंटीबायोटिक्स. सर ने एक एग्जांपल बहुत अच्छा बताया है, अजिथ्रोमाइसिन, है ना. कुछ भी हो जाए सब हम लोग, कुछ भी हो जाए, वायरल इन्फेक्शन रहता है मैक्सिमम, हम लेते हैं एजिथ्रोमाइसिन. एक डोज ले लिया है 3 दिन के लिए, 5 दिन के लिए. बट वी आर नॉट अवेयर कि इस एजिथ्रोमाइसिन में अगर एंटीबायोटिक्स रेजिस्टेंस डेवलप हो रहा है, देयर आर चांसेस दैट एजिथ्रोमाइसिन रेजिस्टेंस में जो जीन्स है वो दूसरे एंटीबायोटिक्स में जाएंगे थ्रू ट्रांसपोसर्स. That is very very important as far as antibiotic resistance is concerned. तो सबसे important है खुद से शुरू करना है, avoid antibiotics as much as possible. दूसरी जो चीज सर ने एकदम बराबर बताई है, वाटर में एंटीबायोटिक रेजिस्टेंस ऑर्गेनिजम्स आ गए, प्रिवेंट कैसे करें? So that is obviously not as it, but obviously, sir, administrative level say it has to be a very, you can say, a strict implementation.
Certain elements are required to be done by
yes. उसके बाद हॉस्पिटल इन्फेक्शन कंट्रोल कमेटी होनी चाहिए, which should be सिर्फ पेपर में ना होके, it has to be implementing some things. तो वाटर सर्विलेंस एक फैक्टर है जो हमको महीने में कराना ही कराना है, भले वो अपने वार्ड में के पानी हो, भले वो हॉस्टल का पानी हो, भले वो कैंटीन का पानी हो, जस्ट टू सी वेदर उस वाटर से हमको एमडीआर ड्रग्स, एमडीआर आइसोलेट्स तो नहीं मिल रहे. सो सर्विलेंस इज आल्सो वन ऑफ द इम्पोर्टेंट फैक्टर which can be used for prevention of your this MDR. और लास्ट में जो है वो जो सर ने बताया अभी, एंटी माइक्रोबियल अवेयरनेस वीक, उसमें एंटी माइक्रोबियल स्टीवर्डशिप प्रोग्राम इज वेरी वेरी इंपॉर्टेंट. तो क्या है ये एंटी माइक्रोबियल स्टीवर्डशिप प्रोग्राम? कि भाई आप सही दवाई सही पेशेंट पे सही डायग्नोसिस में सही मात्रा में सही रूट से उसको दे दी जाए. सो दैट एंटी माइक्रोबियल स्टीवर्डशिप प्रोग्राम जो है वो बहुत इफेक्टिवली मेंटेन करना चाहिए. सो दीज आर सम ऑफ द फैक्टर्स व्हिच विल हेल्प अस टू कर्ब द एंटी माइक्रोबियल रेजिस्टेंस. Okay, so wonderful presentation.
Anyone would like to have one small questions? Anybody, one or two? Yes.
My question is that whatever biomedical water is also microorganisms that survive in the water also. Like that you also take the non-veg. Right. It also affect the animals.
Hello.
What is the to move to the deeper from the medical wastewater, because medical wastewater is also important part, because hospital everyone is going to the hospital, and biomedical wastewater also. So how do we reduce the antibacterial resistance to the water, and we have to clean the water. Right. So it is not only source the hospital. There are various other sources also. The poultry is also one of the source for the development of the antibiotic resistance. Because in poultry business, they are using the heavy antibiotics to grow the chicken. And the heavy antibiotic which they are using, that is being fed by the chicken, as well, whatever the waste is being developed, that is also discharged into the environment. That is also going to mix with the water bodies as well to the soil also, and the microorganisms whichever are present there, they will develop the resistance. Because the required quantity of the antibiotics is not sufficient to kill all the microorganisms. Definitely the dose which will be available there in the water, that will be too low, and the bacteria will learn about how to consume it. So this is one of the real problem which is to happen and is happening. How to overcome is, that is the surveillance and monitoring. Means every hospital I am telling, as well such industries which are using the antibiotics for the development of their stocks or the produce, they should have the water treatment technology. Nowadays, the small scale wastewater treatment plants are developed, which help these industries as well the hospitals to do the treatment of the water before they discharge it into the environment. So that is the way how we can regulate the development of the resistance. But in this, who is to participate? All those industry people, including the hospitals, who have set up the units to install the wastewater treatment plant, so that the water which is they are discharging from their unit, that is treated properly, and no of the microorganisms or the antibiotics is remaining there before it is discharged.
So, if anybody, one more question is acceptable, otherwise, no one? Okay.
No questions? Okay.
So thank you very much, sir. Actually, this one of the question raised by him is very important, because biomedical waste management system, we have solid waste management and all. One of the thing is the things which are washed into the sink, blood or other tissues, etc. That also one of the thing. Actually, as you told that wastewater management and biomedical waste management systems have to be implemented in hospitals also. Otherwise, this will be there, and their water bodies are there. This is one of the biggest issue, because in India we have the wastewater or this sewerage water is going into the water bodies, and that water will again come into the drinking water sources, animals and many things they consume, and this will be into cycle. So we need a very rigorous thing, and you have raised a very good points through your research. We are thankful to you and presenting here. Thank you very much.
Thank you, sir, for your insightful lecture. Your perspective on hospital effluence as a pathway for antibiotic resistance dissemination have provided us valuable knowledge on environmental and public health concerns. As a token of gratitude, we request our co-chair to kindly facilitate Dr. Shailendra Kumar with the momentum and registration kit. Thank you, sir.
Thank you, sir. We now move to our second lecture of this session. It is my honor to invite Professor Dr. G.S. Tomar visiting professor, Guru Gorakhnath Institute of Medical Sciences, Mahayogi Gorakhnath University, Gorakhpur, to present his talk on an Ayurvedic perspective on antimicrobial resistance. Professor Tomar, may we kindly request you to